All right, going to my phone for this one. So I'm going to use the whiteboard a little bit. So I was just telling you a little bit about knockout mice in concept. You can skip the actual work here. Not crucially important. Just understand the concept of using an animal model. Uh, using biotechnology to remove a gene, and then looking at the effect on the animal model to learn about the gene. Next thing here is gene cloning and genomic libraries. I want you to read that, but you don't have to worry too much about it. cDNA stands for complementary DNA, and DNA probes. So what I want to say about this is, remember that DNA, of course, is double-stranded, and eukaryotic DNA will have introns in it. So one way to remove introns is to make cDNA. So I want to talk about this a little bit. We got this idea from retroviruses, right? A retrovirus is a virus. I'm going to draw a little virus shape here that carries its genetic material as RNA. So here is some single-stranded RNA, right? And when a retrovirus infects a host cell, like if this is a eukaryotic cell here, if it attaches to an antigen on the surface, injects its RNA into the cell, and this is basically mRNA. And what it does is it also inserts at the same time a little protein with it. This protein we have called reverse transcriptase. And you should be able to guess what that enzyme does, honestly. But retroviruses are really tricky little devils, because what the cell does is it sees the mRNA and it sort of says, oh, this enzyme goes with it. And what does this enzyme do? It transcribes mRNA back to DNA. So what the cell will do is it will, the cells get tricked so easily by viruses. It'll use the enzyme, use the mRNA. It'll copy the mRNA back into DNA. So there'll be T's here instead of U's. And then the cell will often make this double-stranded and insert it into the nucleus, right? And that way the virus has infected the host, just like you should be aware of normally. But what biotechnologists have discovered is, hey, we can use this enzyme to make what's called cDNA. So what is this? So let's say, I'm going to make a separate picture here. Let's say that this is a eukaryotic gene, right? We copy it into a pre-mRNA. That would be exactly the same length as the gene, right? And then without the introns, this was our pre-mRNA. And then if we took out the introns, the mature mRNA would look like this, right? So here's the mature mRNA. That says mature. Sorry, my marker's a little sloppy there. That's the final mRNA product. That would be what goes to the ribosome. Introns are removed. That's why this is shorter. So all non-coding sequences that were here are no longer here, right? So if we take this mature mRNA from a cell and we use that enzyme reverse transcriptase, it copies this back into DNA, right? And we call this cDNA, or complementary DNA, right? So we could make this double-stranded, we could keep it single-stranded. This would be, we could use this, we could insert this. If it was double-stranded, which is easy to make it double-stranded, but we could also use it single-stranded, as I just said. We could insert this into a plasmid, right? That way we have a eukaryotic gene with the introns removed automatically. We can also use this as a probe, right? Like here in this picture it shows, if you wanted to find out, does this DNA here have a certain sequence? Well, you could make a cDNA probe that was the complementary sequence to your gene of interest, and you could hybridize them. You could sort of wash a glass plate with the probe with little bits of DNA on the glass plate, and wherever the probe attached to, you would know that sequence was there, right? We're getting a little bit in the weeds here, a tiny bit, but that's what this section is about here. So do read through that. Microarrays. Microarrays use cDNA. This is actually kind of an online activity. I do want you to do this. I think the chance of there being a question about microarrays on your 45-minute exam on the 18th is very, very low. So I wouldn't prioritize this necessarily. If you're really backed up on work for this class, I kind of let you get away without doing this. But I think just doing this activity here would just help you kind of have a mindset about biotechnology and modern biotechnology that would just be helpful for you.